what I want to talk about is this thing called the membrane potential. I want to talk about this battery charge that this pump creates. And I just want to stop here for a moment and just show you this picture. This is a picture of a neuron, and this is actually a soma. And this is an axon leading away from the soma. And all of these red things coming in, you can see like little tails coming in, and then they kind of are expanding. Those are actually all axon terminals coming from other neurons that are all synapsing into this soma. It was not a stain for you to see the dendrites. It was a stain for you to be able to see the axon terminals coming in and synapsing with this, right? There's such a density of them, you can't distinguish sort of one from the other. And they're even coming in onto the axon. So anyways, just wanted to show you this image. It's pretty cool. When I say like cells branch out into like hundreds of thousands of axon terminals, right? Here's a bunch of axon terminals all converging or synapsing on a soma. Now, this movie. I mentioned this if you read the primer. So this movie freaked me out when I saw it the first time. I actually saw it in Horton Plaza. Oh, the parrots are back. I don't know if you can hear them. In Horton Plaza downtown in the movie theater. And I'd been there. My sister lived kind of across the street at the time before she had a family and everything. We were all young. And I used to go to movies there all the time for a bunch of years. And I went to see this movie. And I walked out of the movie theater, and I was with someone else who had also seen a lot of movies there. And I came down the escalator, and I walked out onto like the deck, and I paused, and I freaked out because I swear there was a table and two little chairs that had never been there before. And in order for them to be there, there had to be like a complete change in the architecture of the wall. And that doesn't make any sense. So I like paused and I looked at it and I was like, that makes no sense. I'm like, my memory must be wrong. And I just kept walking. Now, if any of you have seen the movie The Matrix, there's like glitches in the Matrix where people see things that aren't quite right or don't make sense. But anyways, I go a few steps and the person I'm with like grabs on my arm. They're like, hey, why did you pause back there? And I'm like, nothing. No, seriously, why did you pause back there? And I'm like, nothing. And they're like, why? And I'm like, well, because I said, did you see those chair and two table, the table and the two chairs there? And they're like, yeah. And I'm like, and they said, without me priming them, they said, I don't think those used to be there. And we were both just like, are we in the Matrix? Now here's the thing, the reason why it disturbed me so much. A lot of the movie The Matrix is very, very grounded. It's extremely grounded in science. A lot of science fiction movies don't have a lot of science in they're mostly fiction. So there's a lot of things happening in the Matrix movie that make it seem more plausible to a biologist's mind. So first thing, and I can't get to my chat window. Where's my chat window? How do I even see that? I don't know where it is. Why are they all in the Matrix? Why are the people in the Matrix?
To be batteries.
To be batteries. Why are we batteries though? How'd that happen? I know at least a couple people have seen this movie, or maybe like the third or the fourth or something.
I actually haven't seen this movie, but I've heard of it.
Yeah, you're gonna want to watch it, and you'll find it really disturbing.
The machine.
So okay, so here's the thing, because a lot of you haven't seen it, right? Because it's an old movie. It's even more disturbing the more things that have gone on since the creation of the time of this movie. All right,
I
so
know because like, wait, can I say something? I think like the machine won the war and then using the human to sustain themselves.
Right. So here's what's going on. There's people invented computers. Okay, yeah, check. Okay, computers got smarter and smarter and bigger and more capable of lots of memory. Okay, yeah. And then computers got better designs and programs, ability to fix a lot of their programming, right? Small bits of artificial intelligence where, if something, a subroutine runs wrong, they can do a lot of their own diagnostics and do a lot of their own repair and fix themselves, Sure, that exists. And computers could do, then this is where it starts jumping a little bit further into science fiction, but, you know, Elon Musk and whatnot are telling us, you know, with his cars and everything that we're pretty much here. You know, that computers are really smart and that they, in some cases, are probably smarter than the people that created them. And then this is where the science fiction of the movie steps in. So the computers that we created got so smart that they basically, they became artificially intelligent. And when they would meet other computer programs and they would have compatibilities, and then they would, together they would work on problems, and then they would create new computer programs, and those new computer programs they considered to be their offspring, and they were emotionally bonded to them like as an artificial being, right? And so they had generated all these little offsprings, and they developed this artificial computer world, and they needed more and more and more and more energy. And so eventually people are like, dude, you're out of control. You're ruining the energy and you're ruining our planet. And they're like, we don't care about you people. We need energy. And they were smarter than us and they were more powerful than us now. And they didn't need us to fix them because they could fix themselves. And so they enslaved us. So they took all of humanity and they enslaved us. And what we were doing was we started toiling in their fossil fuel mines, and like they needed more and more energy. Right, so we're digging up fuel from the ground and coal mines and whatever, and we're just burning fuels and making solar panels and doing all these sort of things. And eventually we run out of fossil fuels. And at the same time, we burn so much fossil fuels that there's so much pollution, this little bit, like there's so much pollution up in the Earth's atmosphere that sunlight no longer penetrates. And so there's no longer long time into this matrix time. Right, the Earth has lost all its sunlight penetration, and there's no longer plant life. It's just animals and these machines, and the animals are people. And we dig up energy, and we give energy to the machines, and eventually they ran out of energy. And there's no more solar energy because the plants are dead. Now, first of all, what are people eating? And so we've been enslaved already at this point. They're like force feeding us like dead people. Really gross in its own right. Whatever. But now they run out of energy. So they turn to the last source of energy on the planet. Because they can't survive without their electricity to run their machine, to run the programs. And the last source of energy on the planet is people. And they take people, and I'll show you in a second, every single cell in the human body has a small charge. If you circuit all of those charges together, the human body gives off quite a lot of electrical potential, a giant battery, right? And so what they did was they stuck like a wire into me, like now I guess looking at our screen here, like our screen view, like they stuck a wire into me, right? And then took my energy and then stuck a wire out of me and stuck it into the next person and to the next person and the next person. And they linked all the human beings up into a giant wire system. Like, so each of us is a separate battery, all wired together in a giant battery pack. And they could run their machines 24 hours a day using human beings as battery packs. Here's the problem though. If I told you today, you gotta sit in a box with a wire coming in you and a wire coming out of you 24 hours a day, we'll feed you ground up people slurry. Even if you're enslaved, are you gonna put up with that?
No.
Human beings, we don't put up with that. We'll commit suicide. There's loads of times in history where one group tries to oppress the other and the other group just says, you know what, we would rather be dead than be oppressed by you, right? So human mind just at some point will just go, we won't do that. We'll kill ourselves if in order to kill you, we'll kill ourselves, right? So what did the machine do? Machines are smarter than us. So the machines took us and they hooked our minds up to a virtual reality that we couldn't really distinguish from the real reality. And then as long as we were in this virtual reality, we would just sit and be complacent, right? Wire coming in, wire going out, feeding tube, right? You're unconscious, but you're attached to the thing. And they would basically attach you as a baby, right? And then the moment you're born, the neural net would go on, right? And then feeding tube would go in, and you'd be hooked up, and they would put you in a pod that you could grow in. You'd be in a small pod that you would just slowly grow up in. The whole time you're never awake. You're just in a virtual reality world the whole entire time, so that you're a complacent battery. Now, some people's minds in the Matrix, some people's minds would reject the virtual reality at times. And if you rejected it, then you would wake up, and you'd wake up in this pod. And there's a scene in one of the movies where as far as the eye can see are these giant like telephone pole things, but they're big like redwoods, but bigger. And then attached, and like there's pods attached to all of them, like going up, and it's like people. And then it's as far as the eye can see in every direction, like poles and people. And if the mind rejected the Matrix, you were no longer good as a battery. Now you were people food, right? So then the pod would open up and it would drop you out, and then a machine would come and grab you, and then they would grind you up into people food, right? To feed all the other batteries, people. That's the baseline of the Matrix. So you go, well, that's freaking crazy, right? You know, or is it? Now see if I can. I'm so lost in my screen sharing that I can't get I've got too many windows open. Let me see what I can close. My PowerPoint. I need this screen. All right. So, have you guys ever done this? This is a potato, and you take one type of metal, two different types of metals, and you put it in one end and you put it in the other end. We're a charge of, we are, every cell this potato is made up of is a little small potential like a battery, right? And if you put different metals, the battery will be drawn like, just like it is in a battery. It has a positive end and a negative end. It's like two different metals usually on the either ends. And what'll happen is the electrical charge will get drawn from one end to the other. When you hook it up into a circuit, you put a wire come from one end and a wire to the other end, and you attach it to, say, for example, take a wire and you attach it to this light, and you take the other wire and you attach it to this light, you would light up this light. Well, what's interesting about this was I made this picture a long time ago, long before I was a teacher. And this light bulb is actually an analog light bulb, not a digital or LED light bulb, and it takes quite a lot of light to light it up. And when I did this experiment, I was making it for a talk I was doing, and I put in the two nails and I hooked it up and I took the pictures, and this was before digital photography. So I had to wait like a week to get, maybe a couple weeks to get the pictures back. And when I did, the light bulb had lit up, but it was so dim, it didn't show up. So I couldn't use it. But I still wanted to show the effect of the battery. So I said, well, I'll hook two hook two potatoes together. So I took, one color, the same nail. So I put one nail, hook it in, another nail, then I hook this nail to this nail, right, this, that. And now it's two potatoes are running into this light. And when the charge of two, like I turned up the voltage, when I hooked up two potatoes instead of one, the bulbs got light enough that it lit up, right? Took the picture, got my light, did my talk, whatever, years ago. I forgot about it. When I started teaching, I wanted to talk about this, and I wanted to say, every cell has a charge, and you can cause that charge. This cell can't release it. A muscle cell and a neuron can actually release it. We have to put a nail in here and have it released down a wire, right? The cell itself, the potato cell, is not capable of it. But our cells, our neurons and muscle cells are going to be able to discharge this buildup, right? And we won't light a light bulb, but it'll be enough to send a signal from one end of a neuron to another. And again, every single cell in the human body, plus prokaryotes, plus archaea, all have this sodium potassium pump, and they all have a charge, right? It doesn't matter what kind of cell it is. This is like the round happy cell we've sort of been studying. Some of you have had anatomy. This is like these are the types of cells you would see that line our digestive tract, for example, with lots of cilia and microvilli and things like that on the surface for nutrient absorption, in this case, microvilli. These are like fat cells. These little tiny things that are surrounded by all this wavy stuff, these are actually bone cells. These look like champagne bubbles, each one of these little bubbles in the champagne. These are actually cartilage cells. This is actually a muscle cell. Maybe it's got more than one nucleus. right? What all of these cells have in common, one of the things they all have in common is they all have this sodium-potassium pump, and it's all creating this battery charge, right? So I want to talk about that battery charge. And in order to do that, I want to talk about some relative thinking, that terminology that biologists use. So this is not like any kind of trick question, right? So I've got two piles of money, pile A and pile B. And here's one stack. I put it in pile B, and I got one stack, two stack, three stacks. All three stacks are identical, so it's like it's not an optical illusion or something or anything like that. So we asked the question, right? Which pile contains more money, pile A or pile B?
B.
I'm gonna say A.
Gonna go with. Is that your final answer, right? I'm gonna have to go with A, right? Which pile contains more money, right? And your eyes aren't playing tricks on you, right? A has more money in it, right? Now, here's where like some terminology that we use in biology or engineering, right? And it's different from maybe the way you were introduced to these words in math. And that's why I want to point this out. So we'll say like, oh, pile A has more money, so we'll give it a plus sign. And then we'll say pile B, this goes against accounting too. We'll say pile B has less money. So in biology, and sometimes in chemistry, we'll designate there's less of something. We don't use the less than sign. Instead, we put a minus on there. So we've got more pile of money in pile A, less pile of money in B, right? So we'll say pile A has more, pile B has less. We don't mean less negative, like there's no money in there, but that there's just less money. So we give it a negative sign. It's less than, less positive. So less positive is negative in relative thinking. Right now, this is what's going on in our cells, right? Because we have this ion distribution, right? We already know there's an unequal distribution of ions. Right, we already learned this sodium potassium pump is running, and it's pushing more sodium out, and it's pushing the sodium out, right? So it's got more sodium out, and it's got less sodium in, right? That's just a natural phenomenon we're going to be seeing. There it is. Right, so we've got more sodium on the outside, less on the inside, because this pump is constantly pushing it out. Anytime some sodium gets in, it's on a molecule, it enters the cell, whatever, it's constantly just extruding it 24 hours a day. As long as you're alive, it's pushing that sodium out, right? But then the pump is doing more, right? It's pushing potassium in. So it's pushing potassium in. And so we saw there's more potassium on the inside than the outside. Okay, fine. This gradient, the fact that there's more sodium out and more potassium in, this will be interesting the way the cell can capitalize on it, but I want to talk about the battery here. And here's where we got to talk about some relative thinking. Sodium has a positive charge, one positive charge, plus. That's why we write it plus. Some atoms can have more than one plus, like plus, like calcium. It's like $2. So you think sodium's like one plus, $1. Potassium's also one plus. You can think of them as the same. Like a sodium could be a dollar bill, and a potassium could be the single dollar coins. We make it in the US, we have $1 coins. We don't use them that often, but we have them. They use them at the post office. So you can get like mail, postage stamps out of their things that cost a lot of money. Right? But they take $1 coins, right? They're equal in value. But you can only put the dollar coins in machines that take coins, right? And you can only put the bills in machines that take bills. But they still have the same value. They can only go through certain holes or things, but they have the same value. Now, here's the thing. While we've been pushing sodium out, potassium in, we haven't been doing it equally. We haven't been going, one potassium in, one sodium out, one potassium in. We haven't even been doing two potassium in, two sodium out. Well, we've been doing the two potassium in, but we've been going two potassium in, three sodium out, two potassium in, three sodium out. If you start to think about this as like dollar bills, and you start to think about this like the money, pile A is going to represent the outside of the cell. It's going to have more money. It's going to have this accumulation of positivity because you're taking like 3 bills out, always times 3 out, 3 out, right? And inside the cell, you're only putting $2 in, $2 in. You're always going to have more money on the outside than on the inside. So when you go to look at this thing, you're always going to have more positive charges on the outside than the inside. And you can count them, right? 3 out, 2 in, 3 out. Say it's like 2 bank accounts, and I deposit $3 in one bank account, and then $2 in the other, and $3 in one, and then $2 in the other, then $3 in one, $2 in the other. And at the end of the month, I count them. Which bank account will have more money? The one I've been putting $3 in, and not the one I've been putting $2 in each week. They'll both have a lot of money, and we can count them, right? 1, 2, 3, 4, 5, 6, 7, 8, 9. Right, say we have $9 on the outside here. We got 1, 2, 3, 4, 5, 6, 7. We have $7 on the inside. Right, we've got more money on the outside. We have less money on the inside. We've got more positive charges on the outside. We've got less positive charges on the inside because this pump is doing 3 to 2, 3 to 2. Right, so ultimately what's going to happen is we're going to end up with this accumulation of positive on one side and negative on the other. It's going to be separated by a membrane. And I don't recommend it, but if you were to take a battery apart, you would see that that's exactly what you have. You've got like a set of positive charges accumulated on one end, a bunch of negative charges accumulated on the other end, and it's separated by a membrane usually. And then when you put in a circuit, you're connecting the circuit, and for the things to get from one side to the other, they have to cross through the circuit to get to their other side. Right? So that's a cell. Our cell is a little battery. That's why a battery has one side that's the plus end and one side that's the minus end, right? And in order to charge this battery, we have to put those charges there. In order for our cell to end up with this charge, something had to put that charge there, and that's this sodium-potassium pump. And it needed energy to do it. Just like it takes energy to charge up like a rechargeable battery, you can't just do it. You need solar energy or electric energy to recharge it. Our cells run on ATP. So it uses ATP to put those charges out there and push those charges in and accumulate them and make this membrane difference. Right? When I saw that movie, and I knew that every one of our cells was a battery, if you hooked all our batteries together, you could make a lot of very powerful, you could run a lot of machines. And virtual reality was already sort of out. That movie really started to bother me. So in the movie, the first movie, you got to watch it. There's going to be a scene where Keanu Reeves' character, Neo, they ask him to take a pill, take the red pill. And there's a big deal about that that's because it wasn't in the Matrix programming, right? So the machines are running the Matrix programming. And the humans are in there trying to find people and still trying to fight against our overlords. We're still fighting back. And we're trying to find the minds that are receptive. So that's not part of the programming. So if he were to take this pill and swallow it, it's basically saying his mind is rejecting the programming completely. So the moment his mind rejects the programming, what's going to happen is the pod's going to open up and he's going to become people food. But Neo's people are going to come and scoop him up and take him away before the machine can come get him. So, anyways. A long time ago, Dr. Golgi and some other people, they began the ability to study cells. And this is going back into this in even 1600s. So we barely knew what cells were, but they had found animals, microscopes, and they had found big cells like fat cells and nerve cells and skin cells, and they had been looking at them under the microscope, and they had been resecting them. They were keeping them alive in fluids, and they had developed microscopically small instrumentation that you could stick inside a cell, which is amazing. Hundreds and hundreds of years ago, it was the simplest thing to do, by the way. You take glass and you heat it, and you put a clamp on it, and just right when it starts to get molten, you begin to pull. And it'll stretch and stretch and stretch until it gets microscopically thin and breaks. And that microscopically thin tube is actually small enough to stick inside a cell. And that's what they did. They stuck a tube inside the cell. Glass conducts electricity. Then they hooked it up to a wire. Imagine this is like a potato, right? Then they took on the outside of the cell, because they want to measure the difference across this membrane. So they put one wire on the outside, one wire on the inside, and they're going to measure the difference across the membrane. And the inside of the membrane is less positive than the outside. And it's just the way they hooked it up the first time when they first had millivolt meters. They could have hooked it up the other way on the voltmeter, but it's just what they did. So when they hooked up the millivolt meter for the very first time, they discovered there was an electrical readout. There was actual charge. Don't let the fact that this says minus 70 fool you. It's a fully charged battery. It just means that it's fully charged and it's charged up where there's more positives on the outside than the inside. And that means it's fully charged. If we were to stick this probe in and take this probe out, it would flip and go and read positive 70. Right? It just happens to be the way they hooked it up and the way they did voltmeters. Right? So this thing's measuring electricity. That was a big deal to see that a cell had electricity. And if that's all we'd ever discovered, we wouldn't really made much progress. But they would record them, and different cells had different steady readouts. But always the same. Even if you tapped on it or did stuff, it never changed. If this was a fat cell, it was always minus 30. It was always going to be minus 30. Maybe you put a neuron on there, it was minus 70. It was always going to be minus 70. An accident happened, right? We call this difference that we're recording that a cell membrane might have, we call this the membrane potential, right? Every cell has it, right? This is the charge. This is caused by the sodium-potassium pump, right? It's creating an electrical charge, right? Positive outside, negative inside, that's an electrical charge, right? You now know it's caused by this unequal distribution of ions, right? Where it's taking 3 out, 2 in, 3 out, 2 in. And so that's creating the plus on the outside and the minus on the inside. And right, all cells have it. Then there was an accident in science where there was a scientist and somebody bumped his workstation while he was doing it and the voltmeter moved. And he happened to have a nerve cell on the microscope at the time. If he had any other cell, he wouldn't have made the discovery, but he happened to have a nerve cell and he tapped it and the voltmeters moved rapidly, crazy. And then he tried a fat cell and other cells, tapped the dish, nothing happened. Then he put a muscle cell on and it moved again. And he did a bunch of experiments and concluded that somehow muscle cells and neurons were able to change their electrical charge, right? So all cells have this membrane potential, but muscle cells and neurons seem to be able to change their membrane potential, right? To turn it into some sort of action, right? And by the time we knew about electricity, Ben Franklin and all that times, we knew that people could get shot by like a lightning bolt, which was electric, right? And we knew that it could short circuit their brain or stop their heart, right? We knew heart was muscle, could make their arms jolt, right? So we knew that something was going on with electricity. So we finally had found electricity in the cell for the first time, right? Just hooking it up to these voltmeters. And then the accidental discovery of discovering that muscles and neurons could alter it, right? And so there's different types of muscle. You guys don't need to learn it, but there's like the muscle that we find in our heart and the muscle that makes our bones move, our skeletal muscles. And the smooth muscle that we find like lining our tubes of our body, our digestion and our lungs and our blood vessels, right? They all have smooth muscle that allow them to change diameter, push food through. And what makes muscle is they're able to contract. But before they contract, they have to send a message from one end to the other that tells the muscle cell to contract all at once. And they have to send their message quickly using electricity. Neurons also have an electrical charge just like other cells. They have a different problem. They're really long, and they need to send a message from one end to the other really fast so it can keep communicating, right? Muscle cells have to spread the message around itself really fast so all the muscle cells within a group can contract at once. So you get a muscle contraction. Imagine if it was just like one muscle cell contracted, you just get a flinch, and then another one a flinch, another one a flinch. You have to get all the muscle cells to work together to get the muscles to move something, right? So to get all of something to communicate together quickly, muscles evolved to use electricity. And right, for neurons to send their messages from one end to the other quickly, right, they evolved this ability to capitalize on this electrical charge that's in all cells, muscles and neurons are able to capitalize on it and change it. And that's what I want to talk about today is this thing called action potential. So when we look at a muscle cell or a neuron, if this cell was a muscle cell or a neuron and we hook it up to the voltmeter, it would tend to read out around -70. Some cells sit around -90. The fact that the number -70, -90 isn't really important. The fact that it sits negative is. The fact that it has a constant. Whatever cell it is, every time you measure it, whatever it's resting at, it'll always be resting at the same millivolts. One might always rest at 90, it'll always be at 90. Another one might rest at -70, but it'll always be resting there. Not doing anything, not moving, chilling, like all the other cells that have their membrane potential. Now, this potential can change if this cell was a muscle cell or a neuron. And so if we come and we jiggle and shake the dish a little bit, what'll happen is really quick, it'll shoot up all the way to +40. If you blink, you'll have missed it. And it'll shoot back to -70 in less than 1,000th of a microsecond. I take that back. 1,000th of a millisecond. 0.001 milliseconds, right? It'll shoot up. Let me go back. Right? In 1,000th of a millisecond, it'll shoot up and back. When they saw that thing, they had to name, like when the voltmeter is moving, they had to name that motion because mostly it sits constant like all the other cells. But then for brief moments, it shoots up and back. So they had to name that brief moment of activity, and they use the word action to move, like just like in a movie, you go action, and everybody doesn't freeze. They didn't call it the freeze potential, right? So the action potential, meaning it was like the membrane potential was like when it's just sitting, resting, right? And then all of a sudden it acts and it moves. They decided to call that the action potential. And only muscle cells and neurons can do action potentials. And it's defined really specifically. It's this rapid, which means fast, alteration, change, a really fast change in the membrane potential, right? Where it's going to start at its low at like -70, it's going to shoot up to a +40 and back down to -70, all really fast. I've got a typo here. It says a millisecond, but actually it's 1,000th of a millisecond. And it took like another 100 years to figure out like how the cell could make the changes. They thought it had to do with electricity or electrons. Electrons are negative, so they were focused on ions that were negative. They weren't focused on sodium and potassium, which were positive, so they missed it for like 100 years in research until somebody focused on sodium and potassium. And anyways, we now know after a lot of research that this action potential is going to be caused by movement of sodium and potassium ions. And actually, the movement we're talking about is going to be actually diffusion. Sodium ions and potassium ions are simply going to diffuse. They're just going to spread like a fart. And when they do, though, they're going to be able to create electricity in the cell. Okay. And this is why, let me flip back here real quick. This is important. The ion concentration. Understanding, right, that there's always going to be more sodium outside the cell because the pump has been putting it there, and there's always going to be more potassium in the cell because the pump is putting it there. Now, you guys know, and at this point, the potassium and sodium, they are polar, they have a charge. The membrane, the tails of the membrane are nonpolar, they're uncharged. So these sodiums and potassiums, potassiums would love to diffuse out, but they're going to get repelled by the membrane. The membrane is hydrophobic, right? The charge, it repels charges. Say the sodium is going to try to diffuse in because it wants to go in down its gradient, but it can't. It's going to get repelled by the tails that are nonpolar. So occasionally some will slip through, but the pump is constantly, pushing them in the opposite direction and always maintaining these gradients. right? So a nerve cell and a muscle cell will always be sitting here charged, ready to go. And there's always going to be more sodiums outside and more potassiums on the inside. So let me go forward. Any questions? And I cannot see my chat window. Let me find my chat. I can't find it. I don't know. It's like more maybe. There we go. July 9th. Yeah, we have a test Thursday. All right. Let me get back over here. So this change, this minus 70 to positive 40 and back again, we can actually record it using equipment. It was first done long before there were computers. And when they do, they get this image, right? And here's the cell, it's just chilling at rest. And actually we say in muscle cells and neurons, it's membrane potential because it's capable of becoming an action, a movement. We say that its membrane potential is called a resting membrane potential. And if you just use the word membrane potential, you don't know if you're talking about a muscle or a neuron. All cells have it. As soon as I say, oh, a cell's at its 70 resting membrane potential, now you know, oh, muscle or neuron, it's at rest because that means it can act and move. So here it is, it's minus 70, it's just chilling, it's at rest. Then we stimulate this neuron. So like right here, we're gonna give it like a stimulus, which we're gonna learn in our nervous system will be neurotransmitters are gonna come and bind onto the receptors on the dendrite, and that's gonna be a stimulus. And it's gonna change the neuron. And we're gonna see it shoot up on the inside. We're measuring the inside. It was negative on the inside, but now all of a sudden it shoots up and it becomes positive on the inside, right? It goes all the way to positive 40. And then on the inside, it goes back down. It gets negative and negative, and then it actually gets super negative. We draw a line straight across here. You'll see it's actually lower than normal. It's actually below minus 70. And then it levels back off and it goes to rest, right? And we call this big change from the moment it goes up, down, and levels off. Again, that takes 1,000th of a millisecond. And we call that change from negative to positive, right? And then back again to minus 70 to positive, down, and then back again. That is the action potential, right? Minus 70 up to positive 40 and back down again. That is the action potential. When we graph it, that's what the graph looks like. And on this axis, the x-axis, we're measuring time. Right? And as time is going on, it's getting positive, and then as time continues, it's getting negative, and then it levels back off. And on the y-axis here, we're measuring millivolts. And we're seeing it sitting at minus 70 millivolts, and then it's getting positive, shoots up to 0, and then all the way up to positive 40, right? And then it's going down in millivolts, right? And it's coming down. And we just call this an action potential. Now we have words for describing when it's going from minus 70 up. We call that depolarization. It's polar, it was charged. There was a big difference between the inside and the outside. When the inside becomes positive, there's no longer a big charge, a big polar difference. Now it's been depolarized because now inside and outside are both positive, right? So as it's going up, it's depolarizing, it's undergone depolarization, it's now depolarized. Right, then when it goes back down, it's going to be becoming polar again. So we're going to repolarize. We're going to repolarize. It's undergoing repolarization. We're going to keep repolarizing as it goes down. And when it's sitting up here at the peak, they call that the peak, like the mountain peak. The peak of potentials peaks at 40. This one peaked at a peak potential of positive 40. Then it repolarized, it's undergoing repolarization, right? It's going down. Now there's this moment where it dips extra negative. And we have to describe that where it's more polar than normal. It's hyperpolar, it's more negative than normal. So they call it, in this region, we call that hyperpolarization, right? And then it levels back to rest. Again, on this end, this is the resting membrane potential. Then we're back, resting membrane potential, we're back. Those are the terms that we use to describe what's going on in a neuron. Now, there's no math prerequisite on this course, and we're going to look at graphs a little bit, but you can understand how drugs work without being able to graph this. But I do want you to be able to learn the vocabulary so that as I'm talking, hopefully you understand what I'm saying. So if I say a cell depolarized, you know it went from negative and it became positive. But maybe this graph isn't good for your learning style or for your developmental stage. So here's some other ways of looking at it. So imagine you've got a cell and there's more sodium outside and more potassium on the inside, and it's sitting at rest so that there's more positive charges on the outside and less positive charges on the inside, and it's just at rest. Now we already figured out that these ions would like to diffuse, but they can't because they can't get through the nonpolar membrane. So if we were to able to punch a hole in this membrane, like have a protein maybe that could just open up, one that was sensitive to these millivolts, what we call voltage sensitive, one that would stay closed all the time and then only open up when we want to change charge. And it would open up and then close in response to changes in charges. And this exists. Right? This is your cell sitting, and we call this the resting membrane potential. Nothing new here. Just add the word resting. Muscle or neuron. Now imagine that there's a protein that could change shape, a facilitated diffusion protein. And when it changes shape, it's closed most of the time. But when it changes shape, because it's voltage sensitive and it opens up, ions can flow through it, but only the sodium ion. It's got a special shape that only the sodium ion can go through. I've got a visual for that. I made my sodium yellow, so for my visual people, there's our gradient. I made a little protein here. Proteins have three-dimensional shapes, right? They can maintain shapes. That's what makes them kind of unique in the molecular world. So here's this box, it's like a channel, it's closed. Now, if we come and we stimulate this nerve or this muscle cell, this protein is going to change shape, and when it does, it's going to open. And when that tube or whatever opens, right, sodium has been wanting to it's been wanting to move in. It's been wanting to move down its gradient, but it was repelled by the membrane. Well, now there's a passageway through the membrane. This is this is what they call a voltage sensitive. So we'll see that when neurotransmitters bind, they change the membrane potential, and it causes these things to open, right? Well, anyways, the sodium channel is voltage sensitive. It opens, and then it causes sodium to go in. And there's lots of sodium on the outside, so the sodium's going to diffuse in. So it's like taking all the bank account money and start depositing it into the other bank account. Right? So all of these positive charges, we were positive on the outside because we had more charges on the outside. If those positive charges start going in, we won't maintain less positive charges on the inside. It won't be negative anymore. As soon as those positives start going in, we were at minus 70. As soon as one sodium goes in, it's going to rise. Another sodium goes in, it's going to rise. It doesn't take more than a few sodiums before you actually get all the way to positive 40. It's like a seesaw, like the child's thing. And it's like I've got a long seesaw, and it's just barely tipped. So there's a little bit more sodium on one side. But as soon as sodium starts traveling to the other side, right, it quickly doesn't take much before it shifts. And so that's what's going to happen. Sodium's going to go in, and when sodium goes in, it's going to bring its positive charges with it. And the inside's going to become positive. That's what we call depolarization. The energy for this diffusing in, it was created by that pump a long time ago. The pump created this gradient, and now there's this pent-up energy of sodiums that want to go in. They'll just diffuse in, and the cell doesn't have to spend any energy. It'll just diffuse in, and it gets positive. It shoots up to positive 40. We call that depolarization. Now, this protein is voltage sensitive, so almost as soon as those sodiums go in, it senses that, and the amino acids that they're made out of on this side, they sense that positive, and they actually close. And now this channel can't let any more sodiums in, and we're positive. But there's another channel and it's a potassium channel. And when the potassium channel is voltage sensitive, so it's been sitting closed. But now we're positive on the inside, and when it got positive on the inside, that actually opened, the potassium channel opens. So we were positive on the inside, but now that sodium channel closes, no more sodiums can go in. And now this potassium channel opens, and we've got this excess of positive charges on the inside. That's why we're positive. We depolarized. And now we want to repolarize, right? We need to get rid of, we're up at positive 40, and now we need to start going back down. And the only way we can go back down is to let the potassiums, they just diffuse out, right? There was more of them inside. If we open a hole, they just start diffusing out. And it's like spending money out of your bank account. And you had a lot of money in your account, you start paying the bills, it's going to start going down. So as you start spending the money, right, the potassiums are going to flow out, and now you're going to get, your bank account's going to get spent, and it's going to get negative on the inside again. And that's what we call repolarization. Again, the energy came from the pump. It established this gradient a long time ago for potassiums. So now we open it, and there's this pent up energy, and the potassiums just want to diffuse out, like spread like a fart, right? The sodiums want to diffuse in, potassiums want to diffuse out. That's repolarization. Now, the way these pumps are timed, and it's not by accident, the potassium pump, sodium pump gate. The sodium channel opens, the sodium gate opens, sodium goes in, and it closes. Potassium channel opens, potassium flows out. Potassium channel stays open a little longer than the sodium one. So what will happen is a little extra sodium, one more maybe potassium sneaks out before the gate closes. And when one extra potassium sneaks out, we lost one more positive charge from the inside. And when that happens, you're going to get extra negative on the inside. That's hyperpolarization. But then the potassium channel is voltage sensitive. It feels that negative charge, and it closes. And that's what's going to be going on in neurons. They're going to open, and sodium's going to flow in, and then that's going to close, then potassium's going to open, and the potassium's going to flow out. A little extra flows out, and we get this hyperpolarized stage, and then it's going to level back off. The cell fixes it, right? That's what an action potential is. So here's a visual for like people that don't want it mathy, right? You're like, you saw the graph, and you're like, I don't like the graph. I don't like graphs. Things going up, that's depolarization. Things going down is repolarization. It's like, you're a math mind, you're like, this is great. I love this. Maybe this image is better, right? Now, my read-write learners have been uncomfortable this whole time. They didn't like the graph. They certainly don't like this picture. They're like, what am I supposed to do with all those colors and blobs, Professor Shapiro? There's only like 4 words on this whole page. So here's for my read-write learners. And since some of you are multimodal, right? Here's it in words. What an action happens during an action potential. Door. I use the word to describe this voltage sensitive channel. We can use the word channel, door, gate. I've heard it described as a window, right? But the idea is something that can open and close that allows things to flow through, right? So you got this idea, the sodium channel or gate, right? The sodium door is open, right? And sodium rushes down its gradient. Cell becomes positive. That's depolarization. Sodium door is open. Sodium rushes into the cell. Cell becomes positive. We call that depolarization. Now the sodium door closes and the potassium door is open. Now the potassium charges out, right? Cell becomes less positive. That's repolarization. The potassium door stays open a split second longer. I shouldn't say too long here. A split second longer, and the cell becomes extra negative. We call that hyperpolarization. So you can learn this in 3 sentences. You can learn this in 4 boxes. You can picture a graph. Right? Well, if you learn all of it, you're at like the A money level, right? But you can be able to answer a lot of questions about it. Now, in order to understand this stuff, I would have to say, especially with the pace of this class, you do need to understand what a cell membrane looks like, understand that it's selective. Some things have trouble going through, some things easily pass through. You need to know what facilitated diffusion is, right? Diffusion and facilitated diffusion. The idea that if there's more of something on one side, if they can get through, they will. Something needs something to go through, like an aquaporin or something. In this case, we've got these voltage sensitive proteins that can be put in the membrane. They change shape, they let an ion through. The idea that there are atoms that can have charges, like the sodium can have a plus charge. The potassium has a plus charge. We haven't mentioned, say, for example, but chlorine has a different charge, a negative charge, that we just call those charged atoms or ions. And the idea that there's active transport, in particular, the sodium-potassium pump, like, that's why I sort of was reviewing that again last class as well, is if that material lagging on it, it might hang you up on your ability to get stuff forward. I'll talk about the test, I guess, as we get to the end here. I see there's some stuff in the chat window about that.